here has seen Inception? Okay, great. So I was either gonna spoil that or Infinity War, so that's a good sign. But yeah, no, it's awesome being here. This is actually something that I'm super nerding out about because it's something that since grad school, I've always wanted to just give the intro to the movie Inception. It's one of my favorite movies. Christopher Nolan's the man, Hans Zimmer's the man. They're great at when they do their dynamic duos in movies like this. So there's a handful of things that I wanted to talk about in like 20 or so minutes. Most of it is just the science behind false memories, right? And the science behind trying to manipulate memories. What can happen, what can't happen, and where we are. So actually, there's a funny story. Today or actually a couple of days ago, and today, I was digging up some slides on an old course that I used to teach on neuroscience and Hollywood. So there's a lot that Hollywood actually gets right. There's a lot that Hollywood gets wrong. There's a handful of things it gets right with like amnesia and memory. Finding Nemo and Finding Dory is one of the best examples. Inside Out is amazing if you want to cry like 9,000 times during the movie. But Hollywood, they do their homework. And then Inception, there is a lot about implanting ideas that actually isn't that far-fetched. How they do it, maybe, but the idea behind it is maybe a possibility. So when I was digging up some of these files, I actually found one of my former colleagues actually made this graphic where they, it was flattering to be Leonardo DiCaprio in this picture. I have no Photoshop skills, but this is just a quick shout out to the team that actually made one of the projects that I'll talk about, which we actually called Project Inception, a possibility. So it was very much inspired by the movie. So the quick backstory is that there's a lot of, if you're in science or not, there's a lot of different like hypotheses behind why false memories are important and why studying them is important, especially when we start talking about them in the court of law, for instance. But in the grad school days, it really was this like, it's just cool. It's just awesome to be able to go into the lab and say, we want to try to create a false memory, and how do you go about doing it? So how do you go about doing it, right? Like, where would you actually start? Like in Inception, there's that like box thing, and they kind of press a button and it makes an air noise, and boom, you can do it. It's a little bit more complicated than that. But I mean, if we wanted to go and ask like, how could we make Inception possible, right? We got to start somewhere in the brain if we want to start from a scientifically tractable angle. So one of the things is, me and the lab study memory, and memory is really inherently tied to the movie. So how would we go about and try to manipulate a thought or try to manipulate a memory? Well, we can start by asking, I'm thinking right now about a couple of days ago when I found that old slide, and I have this memory of going through my computer files, where I was, what I was doing. And when I'm thinking about that memory, you can say that, well, we know that there's stuff that happens in the brain, and that somehow we can go in and measure that stuff. What is that stuff? Can we manipulate it? So where would we begin, right? We know that memory has a physical basis. We know that it exists somewhere in the brain, but where, right? You have 86 billion brain cells, 100 trillion synapses. That's a big number. So it's a complicated problem, but one I think that does have an answer. So what we did was we kind of took a nugget of wisdom and started reading the classics, right? So one thing was, we stumbled upon, in this case, a lot of papers by Canadian neurosurgeon Wilder Penfield, who I didn't know Canadian neurosurgeons look like buff football players, but apparently they do. So he was a neurosurgeon, and what he would do is he would treat patients with epilepsy. So to treat patients with epilepsy, for instance, this was in the 50s, you can go in and a patient can be awake during surgery because your brain doesn't have pain receptors. You could pull a Hannibal and touch the brain, you're not gonna feel it. I would not recommend that, but you could do it. And what he would do is he would start giving small jolts of electricity to parts of the brain and ask, does it feel like a seizure is about to start, right? Does it feel like you're having what's known as an aura, which is like when your visual field starts to constrict, and it's usually a sign that a seizure is about to happen. So he would go in and start stimulating multiple parts of the brain and ask, do you feel like an aura is happening? And in a subset of patients, it was kind of remarkable actually, this is one of my favorite footnotes in neuroscience, was a subset of patients would say, doctor, I have no idea why, but I randomly remember walking my dog with my grandma on my 21st birthday. Or, doctor, I have no idea why, but I remember when I was 18, I remember walking my cat and going through the park, and it was almost as if they were vividly hallucinating it. And the area that he was stimulating was this area right here. So you have two of them right above your ears tucked inwardly. And that was something special because it meant that in a subset of patients, when you go and just start turning that brain area on, you might inadvertently turn a memory on. That's amazing because it means that it's almost like a domino effect, that there's these dominoes in the brain that you can knock over that causes the chain reaction that is recollection. So one of the areas that we would know through the last 70 years of research that's especially important, it's an area that, if you've seen the Bourne trilogy, it's the area that Jason Bourne probably has damaged, or Matt Damon. It's an area that, if you've seen, Finding Nemo and Finding Dory, it's an area that Dory probably has damaged. Or in Memento, it's probably the area that Leonard Shelby has damaged. But it's an area called the hippocampus. Again, you have two hippocampi, and they're tucked above your ears inwardly. So it gave us a starting point, right? It's like something about the hippocampus is important for memory. If you don't have a hippocampus, you get these cases of amnesia, like in Memento, or if you want to erase a memory, it might be a good place to start, like in Eternal Sunshine, for instance. So to do that, we have to go in and talk to the brain at the time scale that it communicates on, which is, in scientific terms, really fast. So in this case, it communicates on a millisecond time scale. So a single cell can turn on and off in a millisecond. It's amazing. And to do that, we need a technique that can go and communicate at the brain at that time scale. So just by a show of hands, who here has heard of optogenetics? Great. So opto, of course, meaning light. Genetics meaning, of course, genetics. So optogenetics is this tool from, it's a test question. So optogenetics is a tool where we can go in and we can genetically engineer brain cells to respond to brief pulses of light. So the brain is normally a dark place. It doesn't respond to light, besides maybe your eyes and retina. Everything else, no. But there's an amazing technique in 2005 by a lab at Stanford that showed that you can go in, you can artificially hijack the genetic material of a brain cell and tell it glow or turn on in response to light, for instance. So to do that, I mean, it's almost like a 21st century version of Wilder Penfield's experiments. So this is what a mouse looks like with a laser going into its head. It doesn't hurt the animal. Again, there's no pain receptors. It's very careful brain surgery that works. But it's amazing. So it's a way of going in and shooting light into the brain and asking, can you turn patches of brain on and off, or can you turn memories on and off by using light, for instance? So it really opened up these floodgates of going in and trying to reinstate this kind of activity in the brain. And one of my favorite pictures, for instance, that I've ever taken was, this is actually a cross-section of the hippocampus. So if you ignore human ethics for a second and start slicing my brain in this direction, when you get right above my ears, you'll see a hippocampus on the left, a hippocampus on the right. And then you can put it under a microscope and ask, can you visualize the brain cells that processed one particular memory? So that was a technique that we helped develop. And the idea is that not only could you make those brain cells respond to light using optogenetics, but you could actually find specifically the brain cells that hold on to one memory and force them to glow green, for instance, or force them to glow this like avatar blue color. So it's my favorite picture because memory feels like something that we can't really touch, right? It feels ephemeral. It feels like something that, I don't know, it has no substance, right? But the reality is if you have a small enough microscopic finger, you can go in and not just see what a memory looks like in the brain, but again, try to use techniques like optogenetics to actually turn it on and off. So this is a small part of the hippocampus. And then the green circles here, as well as the lines, which are just inputs into the cell, are holding on to a fear memory in this case. Or in the bottom here, the cyan blue ones are the ones that we think are holding on to a positive memory. So we started with fear memory, not because we felt like being evil scientists in white coats in grad school. It was because fear is an easy behavior to measure in animals, right? Science is all about parsimony. It's about what is the easiest experiment that you can do to get to the answer. And with fear, it's something, it's a response that's conserved across the evolutionary ladder. And in animals, for instance, in rodents in this case, if they're scared, what they normally will do is they'll run to a corner and they'll stay still and they'll remain immobilized. We call that freezing behavior because the animal looks like it's freezing in place. And humans aren't that different. Like if you were to hear a loud noise right now, or like in a movie, for instance, usually when something pops out at you, your first response is to just freeze in place. Or I don't know, if you're walking down the street and run into an X, right? It's like you have that deer in headlights look of just like, hypothetically, you have that deer in headlights look and you freeze, right? And then fight or flight kicks in, right? And then you decide what to do because freezing is a way to not be detected by a potential predator, right? Okay, so we have what we consider optical leverage over a memory. So what does it look like? So I'll run through 2 experiments and I'll end with some fun examples of how the extreme interpretation is that every memory is a false memory. There's no such thing as a true memory. They're not carbon copies of the past. Every time you recall a memory, they're susceptible to modification, and you constantly update them from like, I remember a birthday party and my grandma's shirt was purple to it turns blue to my grandma was never really there to begin with. Like, it's really easy to warp memories this way for reasons I'll talk about in a sec. But okay, the way this experiment goes is the mouse is placed in what we'll just call box B. And we use our techniques in this case to find the brain cells that hold on to the memory of box B. But in order to give the animal a fear memory, we give them just a couple of mild foot shocks. It doesn't hurt the animal. It's more like static, right? It's the surprising. Like, again, if you were to hear the loud noise right now, you probably form a very strong memory of that loud noise because it could have been something dangerous. So we give them a couple of mild foot shocks in this case, and we find the cells that hold on to that particular memory, and we trick those brain cells to respond to light. So how do we measure if we can actually induce that memory? Is that the next day we put the animals in environment A, very different environment, very different box, very different sights and sounds and smells. The animal has no reason to be scared of this environment because it was never given a foot shock there, for instance. But we can ask, if we shoot light into the brain and reactivate the brain cells that we think hold on to the fear memory, then the animals should freeze, right? They should display fear behavior even though they're in an environment where, technically speaking, nothing negative actually happened to them. So what you'll see here is the first half of the video. This is really important. The first half of the video is an animal without a fear memory, right? That's an important controlled experiment because you could imagine that if somebody put an optic fiber in your head and started shooting light into your brain, you might also freeze, right? Because it's a weird sensation. So we have to rule that out, right? We have to rule that out. So what you see is this animal, for instance, it's running around. This is box B. No reason to be scared of it, for instance. It does not have a fear memory, so it's an important control experiment. And when we shoot light into the brain, something amazing happens, which is absolutely nothing. And that's good, because it means that shooting light into the brain isn't going to freak the animal out. Now, this animal does have a fear memory, and it's in an environment where, again, nothing negative has happened to it. And then when we turn these cells on, you can see that the animal goes into this, what we consider freezing behavior, or freezing posture. So this is a lot of my life summarized in 30 seconds, but I promise you grad school was a lot harder than that. But we went on to do this both with not just, again, fear memories was just because it was the easiest thing to measure, but we went on to do that with neutral memories, and we went on to do that with positive memories. The positive memories are particularly important because you could imagine trying to reactivate positive memories in an animal model of depression, which actually seems to work. Or you could imagine, if we start off with fear, you could imagine trying to turn off a fear memory in an animal model of PTSD, right? Or trying to turn off fear in a model of anxiety. So the reason we start with this is because fear is also one of those things that's inherently imbued in a multitude of psychiatric disorders. And then with positive memories, we're able to do that as well, to ask if that can, in a sense, act as an antidote, right, to particular behavioral states that we think, or that are involved with psychiatric disorders. So, I said that I was summarizing this for like 30 seconds, or in 30 seconds summarized a lot of my life. This is the kind of experiment that when we did it in grad school, there's like a very real what is happening mentality of like you're seeing something that no one's seen before, let alone something that's inherently tied into movies like Inception. And you really do just get this feeling of like, you're like, what just happened? I think it worked. Like for everybody in and not in science, it does not work 99% of the times. So when it does work, it's amazing, right? And it can open up the floodgates for something that was once the realm of science fiction, namely things like Total Recall and Inception. But now it's like you pluck that from the tree of science fiction and you've grounded it into experimental reality, right? So I think that this GIF perfectly summarizes what it means to pluck something from science fiction and turn it into experimental reality. So to end, I want to end with like a couple of fun games to drive the point home about how malleable or not our memories might be. So, okay, this is going to come at you fast. So this is going to be, just pay attention to the cursor, and we're going to go through a handful of words. They'll be on screen for only 1 or 1.5 seconds. And then the idea is it's pretty straightforward. Try to remember them, right? Everyone that just had wine before this is regretting this immediately. Right. Alrighty. So just hold on to those for 2 seconds. If anybody's seen A Quiet Place, that's what it just sounded like right now for the entire movie. And but okay, so if you want to have an existential crisis at night, I recommend looking up the Mandela Effect. So the Mandela Effect, so BuzzFeed, of all places, did this thing of like top 20 reasons why your life is a lie. And the Mandela Effect, it's a very real effect, and it's called the Mandela Effect because a lot of people remember, especially in the early 2000s and about up until 2010-ish, a lot of people remember where they were when they heard the news that Nelson Mandela died in jail. And they're like, wow, I was being in jail for so long, and like, just that's a crazy story. But of course, we know that Nelson Mandela actually died in 2013 and did not die in jail and was actually released. But it's an amazing effect because there are many people that actually do remember hearing about the story of where they were when they heard that he died in jail, for instance. So they named it the Mandela Effect, and I guess we're in a movie theater so with really good music, so I would be doing disservice if we didn't talk about it. But to illustrate the Mandela Effect, so we'll start off with Queen, right? We are the champions, right? Everyone knows how the song ends. We are the champions, we are the champions, we are the champions of the world, right? Does not end that way. It doesn't end of the world, it just ends we are the champions, and it cuts. So most of us remember of the world. I still remember it of the world. Apparently the Mandela Effect is in full force. The other is, if you're Anthony Hopkins, like what are you saying in this scene? Like, hello, Clarice, right? So apparently, and I didn't know this, he doesn't say hello, Clarice. He just says good morning, Clarice. So the hello, Clarice just kind of came out of nowhere, and we all take it as this like cultural myth that's propagated, but when our internal subjective experience recalls the version that didn't happen. So we'll go through a couple of quick ones. Probably the most famous one is Luke, I am your father. This one's a little bit more subtle. He actually says, no, I am your father. So the Luke, I am your father, or the Luke part is never said. Okay, just by a show of hands. So is it Sex and the City or Sex and the City? So it's actually the one on the right. Finally, in a couple of examples that kind of crushed my soul. So the life is like a box of chocolates. Apparently it's life was like a box of chocolates. So that one's a little bit more subtle, but once again, we all say life is instead of life was. One more left and right one. So which one is the Monopoly Man? So it's actually the one on the right. The Monopoly Man apparently doesn't have a monocle. So what psychologists think are happening is that we're actually remembering the Salters, the peanut guy. So the planters, right? So everyone thinks that we're remembering the planters guy, and then somehow we're incorporating the monocle that the peanut has onto the Monopoly Man. And then in one, if you want your childhood crushed, there's the evil witch in this case. It's mirror, mirror on the wall. It's actually magic mirror on the wall. Somebody knows where the mirror, mirror part came from. I forget what it was. This is the irony of studying memory is that mine is terrible. But it's, but yeah, so it's an example. Like all of these are examples of the Mandela Effect in place. And this is, these are facetious examples, and they're examples that don't really affect our daily routines all that much. There are very serious ones, though, and you could imagine, right now it's funny because we misremember a quote or we misremember how a song ends, but just as food for thought, imagine when those details infiltrate the court of law, right? It's like, that's where I think it really matters because, Neil deGrasse Tyson has this great quip about, like, in the court of law, eyewitness testimony, of course, is one of those things that really matters, right? It may not be a golden standard per se because we need things like DNA evidence and corroborating evidence, so on and so forth. But it's still taken, obviously, very seriously. Now, in the court of science, our eyewitness testimony, we almost go out of our way to try to remove that part, right? This is why we have double-blind procedures and stuff, because we want to get rid of our emotional contingencies, right? We want to get rid of our biases about, did you have a cup of coffee this morning, and is that influencing the bar graph that you're showing me, right? No, the bar graph, the data just is. So something to consider. Now, okay, before we move on to the very last part, so raise your hand from the list of words that we saw if you remember seeing this. Right. Raise your hand if you remember seeing this. Okay, raise your hand if you remember seeing this. Raise your hand if you remember seeing this. And finally, these, so there are two that weren't on it. One is easy, one is hard. God was not on it. Beer was. I realize how ironic it is to have these things up there now, but But yeah, so it's an example of like, it's pretty easy to warp our past, right? That's just with words, right? Imagine with experience. So it turns out that, and then I'll end with just the last bit of science, is that when you're recalling a true memory in this case, there's your hippocampus lights up and when you're recalling a true memory. So this is just a human under an fMRI. And when you're recalling a true memory, you get this increase in hippocampal activity. When you're recalling a false memory, you basically get the same increase. So why is that? Like, why is it that the machinery that helps you recall the past also helps you warp the past? If I had an answer, I'd be out of a job, so we don't know, but it exists, and it's a real thing. But that gives us a place to start now, right? Because if we want to try to pull an Inception and create a false memory, why not start in the area that is active when we're recalling false memories? This is the last experiment. The behavioral schedule, we call it, is just a little bit more complicated, so bear with me. But using optogenetics, using the tools that I just discussed before, imagine that we put the animal, in this case, in environment A. Box A, right? It's forming a memory of box A, but the only difference is that in this case, nothing happens here. The animal is just exploring the environment. It's running around doing sniffy, scratchy, mousy things. The memory of that box A is being formed. We're finding the brain cells that hold on to that memory and tricking them to respond to light. So that's it, right? So that's all that's happening in this case. The next day, we'll put the animals in very different box B, and then we'll shoot light into the brain to presumably reactivate the memory of A. But now the key thing is that while we're reactivating the memory of A, we give the animals a couple of mild foot shocks. So that's the attempt, right? The animal is artificially recalling the memory of A, but while it's getting a couple of foot shocks, to ask, can we actually connect those two? Can we shoot light into the brain, reactivate the memory of A, and in a sense, artificially link that memory with the negative information in the form of foot shocks? Because if we can, then the next day when the animal is back in A, it should behave as though it was given a mild foot shock there, even though technically speaking, nothing bad happened there. Does that make sense? Great. So this is the video. So the first third is the animal running around right now. It's in environment A. It has no reason to be scared of it. Of course, it's running around exploring. The brain cells that are active during the formation of this memory are now becoming light sensitive in the hippocampus. So then the next day, for instance, the animal is giving the negative stimulus in the form of mild foot shock. And you can see the light a little bit where we reactivate, presumably reactivating the memory of A. And now, this is the last third of Inception. When we put the animal back in that cage, for instance, it goes into this stereotypical freezing posture. And the same thing works with positive. So we can do not just tie a neutral memory with negative information, but also a neutral memory with positive information. And now once again, to end with the implications of this all, more recently, imagine being able to reactivate a negative memory, but while you reactivate, or imagine that the animal is naturally recalling a negative memory, but then you're able to artificially reactivate a positive memory to try to almost switch that negative memory. So, and that's possible. So these are the kinds of things that we think are important because all of these have implications for things like anxiety and PTSD and depression, things for which we really haven't had any good treatments since maybe the big pharmacology revolution starting in 1949. And then I think now we can try to use some of this to actually not just figure out how the brain works, but how does memory work? And then how can we use this to try to treat certain symptoms associated with these disorders? So with that said, it's just a quick shout out to the lab because I literally wouldn't be standing here without them. They're the ones that are like basically doing all the hard work. I just signed the checks. And with that, thank you very much.